Our speakers today are Benjamin Cole and Mónica Herrero Huerta. As you will likely recall, the way that we organize this seminar series is for each session, we have two speakers. Each speaker has around 20 minutes to give a short seminar, and then we've got time for discussion afterward. The seminar series, Plant-Phenomics Phridays, is supported by HIPS, the high-intensity phenotyping sites, a multi-scale, multimodal sensing and sense-making cyber ecosystem for genomes to fields, which is funded by USDA NIFA and the NSF. Also, IRA, the AI Institute for Resilient Agriculture, funded by USDA and NIFA. Our first speaker today is Benjamin Cole. Benjamin did his PhD research at the University of California, San Diego, and at the Salk Institute, focusing on morphometric analysis of Arabidopsis hypocaudal elongation under vegetative shade. He then began his postdoctoral research at the University of Southern California, studying diurnal and circadian transcriptome dynamics of the mottle grass Brachypodium distachyon, before moving to the DOE Joint Genome Institute to study plant microbial interactions. He's currently a research scientist at the JGI, where his group focuses on functional genomics technology to profile plant and environmental interactions. His talk will describe efforts to use large-scale sequence-based mutagenesis screens to profile microbial colonization phenotypes. With that, I will turn it over to Benjamin.
Awesome. Thank you so much for the introduction. As mentioned, I am really interested in how plants generally interact with their environment. And to sort of study that, I realize I might be preaching to the choir here in stating that roots of plants make a really great model system for not only development, but also environmental interaction. This is not only because they form the interface with a really complex and interesting environment, the soil, but also that they demonstrate a lot of plasticity to changes in that environment. So shown here on the left is a image of some roots grown in the presence or absence of phosphate. You can note that the root architecture is dramatically different, but there are also a lot of complex molecular changes that go on in this response. Roots are also, plants in general, but also roots are display dynamic responses to a wide variety of not only abiotic, but also biotic stresses, including drought and light intensity and heat, but also to bacteria, fungi, insects, and viruses. So we're really interested in exploring this interaction and hopefully identifying some ways that we can improve plant growth, and especially under challenging environmental conditions, which are likely only to be exacerbated by climate change in the future. And so focusing on the rhizosphere, which is this defined as the area around the root systems that's home to a really complex and dynamic population of microorganisms, we have a variety of different niches that those microorganisms can inhabit. There's the overall rhizosphere, which is defined as like around 1 millimeter away from the root surface. You also have the rhizoplane, which is the area that is directly adjacent to the root. So this niche is, encompasses microbial organisms that attach directly to the root system. And you also have the endophytic compartment, which is composed of bacteria that make it their way into the intercellular space of the root, but also fungi such as brascular mycorrhizal fungi that intimately work with plant cells to form these symbiotic systems. And I'll go into AMF in a little bit. And so I mentioned that soil is really complex, and this is because the microbial populations are really complex. Within, I think, a gram of soil, you have up to 10 to the 9th different microorganisms present. And this spans a really broad phylogenetic distribution of bacteria. And these microbes can respond, the environment that the microbes respond to is really heterogeneous. You have the antagonistic interaction among microorganisms as they compete for resources. You have unequal distribution of those resources, such as minerals or carbon sources. You have the presence of plant roots, which offer a not only a rich carbon source for microbes, but also a variety of antimicrobial compounds as they try to select for bacteria or fungi that they want to associate with. You also have different, sometimes unpredictable environmental changes, such as flooding or high temperatures, that make this life in the soil really challenging. So the presence of microbes on root systems make them, in spite of all of these environmental interactions, make them really good symbionts because they are robust to all of these different changes, and hopefully they can provide some benefit to the plant. So plant-associated bacteria are characterized as either pathogenic, which, as you know, they cause disease or reduced yield on crops. But there can also be a lot of beneficial properties of plant-associated microbes, such as scavenging nutrients from the soil to bring them to the plant. They can also make and introduce phytohormones to plant systems to control or influence their growth. And they also can synthesize antimicrobial compounds, which can be beneficial in fending off pathogenic fungi, for example. So one thing that we're really interested in is, you have all this microbial diversity in rhizosphere environments, but what makes the bacteria able to colonize the root, and more so, what makes them beneficial to the plant? And so to really get at this, I'm more of a sort of the reductionist philosophy of biology. One thing we could do is just take a model microbe and mutate every single gene and see what it does and see if it impacts colonization. And that essentially is what is done by this technology called transposon sequencing, or TNSeq, but in a massively parallel way. Coming into this, I, was trained as in Arabidopsis and looking at phenotypes and, designing systems to measure phenotypes. But microbial phenotypes are really, really simple in comparison. You can either have growth or not growth, at least in this system. And so TNSeq works by taking a microbe of interest and just generating a really complex mutant population using a transposon insertion mutagenesis. The way this works is you have a transpose on a piece of DNA, it inserts itself into a gene. But in this strategy, we've tagged these transposons with a 20 nucleotide barcode sequence. So this is a sequence that is unique to the insertion site of that transposon wherever it landed in the microbial genome. And this is a pseudo-random insertion of this transposon into almost anywhere in the genome. Anywhere there's a TA dinucleotide in the genome, this transposon can insert itself there. So what we get is a really rich mutant population. And then we can profile it, either growing under rich media in vitro. And so here, if the transposon has mutated a gene that is essential for any kind of growth, that mutant will drop out of the population. And then when we sequence the insertion site of this transposon, we will get a lack of sequence events within that gene. This will tell us that this gene is essential for this bacteria to live. In addition, we can then challenge the microbial library under a variety of conditions. And here, not only will we see a lack of the essential genes, but we'll also see that the mutants in genes that are responsible for growth under this specific condition also drop out. And we see a lack of that gene as well. And so we can define this gene as required for a different condition. We're sort of profiling all of these mutant phenotypes under these in vitro conditions, but can we apply it to plants? And so to do this, we took a model microorganism, Pseudomonas fluorescens, although it's since been reclassified as a Pseudomonas simiae. due to a closer phylogenetic relationship with that distribution. And it gets its name, or at least it got its name fluorescens, because it's able to glow under UV light or under dark conditions. And then we take our favorite model plant, Arabidopsis thaliana, because we can grow it at very, very high density and get a lot of colonization events, ideally. And then we constructed our transposon mutant library using this barcoding strategy. And the benefit of this barcode, as I mentioned earlier, is that we don't have to sequence the insertion site every single time we profile this transposon mutant library. We can do this once and associate the barcode with the insertion site, and then in subsequent assays, we can just sequence the barcode region, which is much more robust in a variety of different conditions. So we can profile this library over and over and over again, the same library, and compare conditions to each other. And so this is what the overall assay looks like. We take a upgrowth of the library that is then concentrated down and applied to plates. We then take these Arabidopsis plants and transfer them to plates containing the mutant library, wash and grind the roots, and then isolate our rhizoplane sample. We can then take a variety of different controls, one of them being the input library itself, but also we took a small square of this mesh, this nylon mesh, applied it to a plate of bacteria without any plants, and then I harvested that as well. And then we sequence the barcodes that are present in all of these different populations. And what we expect to see is that mutants, genes that have, when mutated, reduce the ability of that bacteria to colonize, will have more or less normal abundance under control conditions, but have a substantially reduced abundance when we see them in the root. In addition, we also expect to see some mutants that have more or less normal control abundance, but also increase their abundance on the root. And these genes may be dispensable or detrimental to colonization. And we can then take this profile and overlay it on a map of the bacterial chromosome to get this genome-wide profile of bacterial colonization on plants. And some of the major features that popped out of this genomic profile were things that you might expect to see. One of them is a canonical flagella biosynthesis set of operons that, if the bacteria cannot move either through the plate or in the soil, or they cannot move around on the root itself, that is a really detrimental phenotype to have on plants, and so it can't really colonize all that well. So we expected this, and it was nice to see that this sort of validates our assay. Some other things that we saw were some mutants clustered in these operons that are putatively responsible for carbohydrate or cell wall biosynthesis metabolism. But we also saw some genes, some sets of genes that are detrimental to colonization, or you mutate them and they grow more on the root. And those include amino acid transport metabolism and some pili assembly. More interesting, we identified around 45 genes as hypothetical, meaning that we have no idea what they do. They have no annotation. They don't really look like anything that we know anything about. So what we wanted to do is first validate the assay and then look at these hypothetical genes. And so one way the approach that we took to validate the assay is to generate a luciferase producing version of the wild type version of this bacteria, which glows under if you put it in a dark box, you can see the glowing of this bacteria on the roots. And we designed a competition screen where we mix this luciferase producing strain with an unlabeled mutant strain that we picked out of our library, and then see how it performs versus the wild type. And so here I'm mixing wild type non-luciferase with the luciferase version at a variety of different fractions. You can see the increase in glowing of the roots, which we quantified just by quantifying how intensely the fluorescence was here. So this shows that the assay is more or less dynamic, that we can pick up some subtle differences in colonization. And so we then selected a handful of genes that are predicted to be deficient in colonization when mutated, and some of those that are that when mutate them, they grow more on the roots, and more or less validated that the genes are behaving as we think they are. And so going back to those hypothetical genes, we turned to because this T-and-seq strategy is highly it can be amenable to high throughput methods. So we don't have to just profile them on plants. We can actually design dozens of in vitro assays and microtiter plates where we vary a lot of different conditions, such as carbon availability, single nitrogen sources. We put in a bunch of stress conditions, such as antimicrobial compounds, antibiotics, and we also profiled their motility using a soft agar assay. And so these 88 different plate-based assays can help us identify functions for these hypothetical genes, where maybe this gene is annotated as hypothetical, but it has a really deficient phenotype when you grow it on a soft agar assay, for example. But we also identified some hypothetical genes that really don't have any phenotype at all. So here are some of the genes that we, some of the classes of things that we found from this this in vitro based method. These are genes that are involved in different carbon utilization phenotypes. These are genes that reside in a putative operon that is responsive to antimicrobial compound synthesis. And then these are a lot of amino acid biosynthesis or metabolism genes that tend to cause the bacteria to grow more on the roots. And so one of these sort of hypothetical genes that we identified from the library, we've sort of started to follow up on this, and a postdoc in our lab now, Janelle Basso, has been really hitting this set of genes hard, as they seem to be related to phage processes. And so we found several loci within the genome of P. simiae that are putative latent phages. And Janelle actually demonstrated that these phages are active, meaning that when you treat them with a chemical inducer that stresses the bacteria out and causes the phage to activate again, you get this clearing phenotype in cultures. And we also demonstrated that the prophage is active by PCR, and we're starting to get some flow cytometry data to suggest that yes, this phage is a latent phage that is activatable, and we're really excited by this because we think that maybe there's another layer to plant microbial association that is dictated by the activity of these phages. That perhaps when a phage or a bacterial strain gets highly dense on the root, or it's stressed by some environmental condition, this phage may be activated and might contribute to the population dynamics in the root. So this is sort of early data yet. We don't really have a lot of additional follow-up, but we're pretty excited by this. So to summarize what I've said so far, we've developed a genome-wide colonization assay using transposon sequencing. We identified over 300 genes that in some way influence colonization, as when they are mutated, they have a different phenotype on the roots. We developed a luciferase-based validation screen, and we identified that carbohydrate cell wall metabolism is really important for colonization, whereas amino acid metabolism and others are dispensable for colonization. The amino acid story we think is because the plant is actually supplying a lot of nitrogen to the bacteria, that they don't really need to de novo synthesize a lot of amino acids themselves, and doing so just kind of hinders their growth profile on the roots. But that's just some speculation on our part. And then we're following up on this bacteriophage lead. So to now, I've sort of talked about roots as a monolithic entity, but we're trying to now delve into the roots as a collection of cell types that we're pretty excited to sort of to go into. And I'll just kind of go through quickly because I realize I'm running short on time here. So one system that we're really interested in looking at is the association of plant and arbuscular mycorrhizal fungi. And this is interesting not only because the association is very important agriculturally, but also because the association is highly cell type dependent. These AMF only colonize a specific set of cells within the root. And so what we want to do is use a suite of new technologies called single cell transcriptomics technologies to profile this association and hopefully identify genes that are really important for the association itself. So to do this, we are using a technology called spatial transcriptomics, which uses an array of barcoded spots on a slide that you then take a frozen tissue section and expose it to get this spatially resolved map. And in parallel, using more canonical single cell dissociative technologies that you may have heard about that can profile single cell transcriptomes. And so this is some really exciting data we've got so far using the dissociative technologies. We've identified a population of cells that seem to be associated with mature arbuscules that we're following up on now. And so I think I'll stop here and thank all of the wonderful people that were involved in all of these projects, and maybe pass it off to Mónica for the second talk, unless there's time for questions.
Thank you, Benjamin. Let's go ahead and do the second talk since the topics both involve roots, and maybe we have a good discussion afterward, including questions. How's that sound?
Sure.
Okay, so Mónica, I hope you're ready. Benjamin, thanks so much for a great presentation. Mónica, would you share your screen?
Yes, can you hear me?
Yes, we can hear you. And while you're getting your screen up, I will introduce you. Mónica Herrero Huerta is a civil engineer with a PhD entitled Close Range Photogrammetry Applied to Agroforestry Engineering from the Department of Cartographic and Land Engineering, University of Salamanca in Spain, 2016. Postdoctoral studies from Delft University of Technology in the Netherlands in the Department of Geosciences and Remote Sensing from 2015 to 2018. She is an active member from the TIDOP research group, which is Geomatic Technologies for the 3D digitization and modeling of complex objects in Spain. Currently, Mónica is a research staff member at Purdue University for the Institute for Plant Science, College of Agriculture, developing close and proximal sensing data processing pipelines to crop phenotyping. Her research interests to date have been focused primarily on close-range hyperspectral photogrammetry and LiDAR by alternative platforms, and specifically in computer vision and deep learning analysis by multi-sensor data fusion applied to plant science. Mónica, we can see your slides right now, and we look forward to your seminar. Thank you for joining us today.
Thank you very much. Good morning and thank you. I'm Mónica Herrero, working from the Institute for Plant Science, and I'm going to present our novel approach to model roots in 4 dimensions. So when I prepared this presentation, my main goal was to be very simple and clear. You let me know if I get it. So let's start with the overview. Roots are directly connected with the agricultural productivity, as well as linked with the relation genetic environment. Climate change, environmental degradation, and food insecurity are challenges for our agriculture. Relying on a sustainable agriculture, we want to help to overcome the plant phenotyping bottleneck and improve as well carbon sequestration by using image technologies. So there are several studies that highlight the potentiality of the 3-dimension of roots. For instance, the photogrammetric platform from UCA, University of Georgia with Alice, and the grid work from Danforth Center using X-ray, or the open SIMROD dealing with functional and structural modeling. So our goal is to develop a novel pipeline based on 4D root data, 3 spatial dimensions plus the temporal dimension, which analyze the spatial and the temporal dynamics. The pipeline should be optimized for high throughput, so should be repeatable and robust. The system we'll use is the X-ray compute tomography, where roots can be scanned inside the pot. And for the validation of our approach, we use this sample, 3 samples from tomato that is on the left, and on the right side is corn. In tomato, we use 2 time slots, and in corn, 3. So there are 2 steps in our methodology. The first one is the skeleton extraction, which drives the correspondences over time. So this step deals with the temporal dynamics. And the second step gets the cylindrical model, dealing with the spatial dynamics. The curved skeleton is a line in 3D that summarizes the topological features from the object. So in that case, we apply a Laplacian smoothing contraction developed by Kim-Chun. We like this methodology because use the original geometry, not any resample data. And the key is to control the contraction by little by little. In this image, I remark 2 zones, just to see how the algorithm gives more importance to the main root than to the ramification. For the second step, the root is modeled by cylinders. And we use the approach of Ramonen from Tampere University in Finland, where they model trees with no leaves. In this example, in this figure, we can see how this algorithm removes the noise. On the left is the root captured by the system, by the X-ray system. And on the right is the cylindrical model. And you can see many inconation, many noise that the model doesn't have. As well, another sample for noise removal is this, and this root is from a tree. And you can see on the left, there are many problems with the cementation. and we get many noise. So at the end, on the right, the model, you see how this structure is removed. So let's see some result. This is the result from the tomato plant, where you can see on the left the X-ray segmentation, and then we do this skeleton extraction, and finally in the right is the cylindrical model. And this is the same for corn. So here in this video, you can see the complexity of the model. It's only 2 minutes, and I think it's worthy to see. Here is just the segmentation. So we have the, let's say, the root already segmented in 3D, captured by the X-ray system. And now we can see the skeleton. And we can see it's not very simple, the skeleton, this line in 1D, in 3D, sorry. And there's the cylindrical model, where we transform, we model the figure, the root extract from the X-ray. You can see the cylinder in different colors. And you can appreciate the main root. So we do this one the same from one corn sample. So here is, let's say, the root extract by the X-ray system. And we can see a little part of noise. When due to the moisture, it's difficult to classify the root and the soil. So now is the skeleton extraction. And here you can see how the ramifications are very well summarized, and the difficulty in the first stage, in the starting point. And this is our root model, our cylindrical model. That is very clear, very clean. Okay, I hope you have got an idea. So, the errors of the model. The first important is the loss of connectivity along branches, as you can see on the left. On the right, you can see the difference in volume from the real digital twin acquired by the X-ray and our cylindrical model. And these graphics represent the volume correlation in tomato in the right and on corn in the right. Tomato in the left, corn on the right. And about the temporal analysis, we use the skeleton to drive the registration between different slots. So in the figure, we have the same root at 2 times. And using the skeleton, we can register both scans. And we can easily compute the elongation rate in 3D. And here in this figure, we use the convex rule just to summarize the growth from the root. So the red one is 1 time and the gray is another time. and we can see how the root is growing. So to end with this presentation, we develop a pipeline to automatically extract trace from roots. And we analyze both spatial and temporal dynamics. Our pipeline has potential advantage. It's fully automatic, it's accurate, and fast and robust, and can provide scalability to a comprehensive analysis of high-throughput root phenotyping. And this methodology may help to understand how actually plants work. And we already have 2 publications, a conference paper and a journal paper that was accepted this week in plant methods. So thank you everyone for your attention, and to take 1 minute, 10 minutes to stay here. And I will please to respond any kind of question. Thank you.
Thank you, Mónica. So we have 2 nice presentations today, both on roots and the substrate that roots exist in and interactions they have with the substrate. So with that, I will turn it over to the audience in the room and the audience online for any questions.
I actually have one question for Mónica. So when you're doing the temporal modeling of the roots, do you model each frame independently, or can you use information from like the previous or the other images in your series to help refine some of these roots and hopefully to limit some of the errors that you see?
So what we are doing for the analysis, the temporal, is just with the skeleton from each 3D model, we extract this skeleton, and with this skeleton, we can find the using PCA, principal component analysis. We register both scans, and we are able to compute this difference. So for us, the problem was to register both scans, because you have one tier, one root, and another, and you don't know how to overlap these 2 scans. So with the skeleton extraction, we can make it very accurate, because this skeleton extraction gives more importance to the main root than to the ramification. So you can overlap both scans.
Mm-hmm.
I don't know if I clarify your question.
Yeah, I think so. Thanks.
Thank you, Mónica. Other questions for Ben or Mónica, or topics that people want to bring up? Here's a question from Colleen in the room at Iowa State.
Thank you. And thank you for 2 great talks. Mónica, I've got a question about the tomato plants and corn plants. Are those grown in controlled environments, or like a greenhouse, or are they grown out into the field and then transferred to a facility to do the scans?
No, they are already grown in greenhouse, but in the scan from the tree, you can see how it's transplanted. But in the root, the root in corn and tomato, they are growing control condition. That's why, let's say it's more simple. There are less. We can control better.
Great, thank you.
Questions for Ben or Monica? Here's one in the room.
Hi, thank you for your talks, guys. I guess I have a follow-up question to Colleen's for Ben, but in any future experiments, are you planning on looking at like changing environments and interactions and how like, you know, night, like day length or day cycles where it gets hot and cold might affect any of the bacterial colonization traits you're looking at?
Yeah, we've thought a lot about this. We don't have any immediate plans at looking at. I mean, one thing we thought about is growing plants in short days or long days influence the microbial population, and at least for these colonization traits. And you think they might because the sugar profiles that are excreted from roots should change between different day lengths. But we haven't tried that. It's something that we considered doing, but you know, we don't have any immediate plans to do that.
Good question. Interesting ideas on how to address it. Anybody else? I see a question online. Is the skeleton extraction code for plants' roots extraction publicly available, Monica?
Yes, it is. The noise on this paper, I can put in the chat the page, the link to the code.
That would be great. Fantastic. Thank you very much.
But one thing I have to say, I make little changes. In that way, the skeleton is working with the roots. I make a little changes.
Thank you. Yes, so please share that link in the chat. And I'll give time for any more questions or discussion that we might have. Oh, good. We have one from Koshik. Hi, Monica. What are the biggest challenges in noise removal?
It's a tricky question. For me, because I just model, let's say, if it's not similar to a cylinder, or if there is no morphological and topological rules that say this is part of the tree, I just remove. But for the person, Valerian, who is doing this segmentation, it's more difficult for them because he needs to try to do better. But what I'm doing is, okay, if I see a mess and the computer cannot understand the shape and everything, it's not working, the algorithm, so the algorithm remove this part.
A comment from Mitch Tynstra. Monica, can you discuss opportunities for integrating root and shoot-based phenotyping over time?
For me, it's clear. Over time, the more difficult part is just to match every ramification over time. This is our next step. We want to be able to, let's say, to matching this ramification, because we just match the full root right now. So the next step will be to match all the ramification over time.
Monica, this is Mitch. My question has to do with your presentation describes your work looking at roots and feature extraction for phenotyping using the X-ray CT. But at the same time, these plants are also being imaged for the above ground part of the plant. That's my question. What are your thoughts about integrating shoot-based and root-based phenotyping in non-destructive mode over time?
Of course, should be the following point to put together, because the plant is only one. And if you can understand the full plant better. So the opportunities are amazing, because we want to try to understand the full plant and the functionality of this plant.
Thank you. Any other questions in the room or online? Seeing none, I will thank you all for joining us today. We have one more Plant Phenomics Fridays in November. That'll happen next week. We hope to see you soon. Thank you again, especially to Benjamin and Monica for the preparation that you've done for today, for delivering excellent seminars, and for giving us just a little bit of time to get to know about your research. Have a great weekend, everybody.